So, I mean, this is my current favorite area of biology, and it really comes from trying to think about our patients and better ways of serving them. So the autologous cells that we work with at the moment been making great strides with changing their biology, making improvements into those cells. But you always rely on the fact that the patient has to come into the hospital, they have to undergo leukapheresis, that all takes time, their cells go away for manufacture. And late stage cancer patients, often they don't have a lot of time. So what we're trying to do with the Allogeneic program is to get off the shelf, ready to deliver samples so that the patients can have their cells the next day rather than waiting for a leukapheresis slot and then personalized manufacturing. So that's not an easy thing to do. There's lots of biology to understand and overcome. But that's really the intention of trying to make a product that's very consistent so that lots of patients get the same cells. You don't end up with a different starting material every time because it doesn't come directly from the patient. It's less invasive for them. It's one less procedure that they need to undergo. But I think the power of it is the consistency in the cells that you make with an iPSC platform and the fact that you can do more genetic engineering. So under our autologous system where we use lentiviral vector, we're limited by the vector size for the number of additions that you can make. So you can put the TCR into your vector, you can put another thing like CD8 or various other molecules in, but there is a limit to what you can cram into that vector. Whereas in a stem cell platform, you can use gene editing and you can do multiple rounds of gene editing. So you can add in the receptor in one go, you can add in, a different CD8 if you wanted to. It wouldn't be the first choice, but you can add different biological receptors in, you can add extra facilities, extra functions to those cells. You can knock genes out, which again is very different. You can actually stop you can modify receptors individually, and you can do rounds and rounds of that in an iterative fashion so that you can add everything in together. But you also get to the stage where every single cell contains every single edit, which I think is important, that it's going to be important for product consistency. But it also allows us to learn in patients. So the first trial that we intend to do will go in with our TCR and some modifications to let the cells persist for a long time. But we'll also be able to see their biology and understand what are the most important factors to address, and then go back and edit on top and make a new bank and go back in. So it's a good platform for learning, so that you can learn from the patients and make the next modifications to improve the products. So I'm really hopeful that this is, a good way for us to go with the intent of getting cells that treat patients, and we'll be able to fine tune the efficacy and also fine tune the phenotype of the cells based on what we discover in those patients when we do our first trials. We've said that we will have 2 allogeneic products in the clinic in the next 5 years. And I am under significant pressure for that to be as soon as possible. So the first one will be with our MAGE-A4 TCR. So we will be able to do a direct comparison between autologous cells that we have going forward in Spearhead 1 with the Allogeneic platform to see how well it compares. So same TCR, and we'll be able to see, does it work as well for patients as our first product, our first generation product, or what needs to be changed? And then the second product that we're going to bring forward is the mesothelin hit. So that's combining that HLA independent TCR with our Allogeneic platform in partnership with Astellas. So we'll be taking that forward together to really test a new sort of TCR, which is very exciting in a new platform, but also having that benchmark of the MAGE-A4 TCR already. So those are the plans, and we're working through a lot of the genetic modifications that we require and getting the scale right for that clinical program. So very hopeful that we'll be in patients in a relatively short period of time.